Okay, everybody, jumping into now chapter 8, smooth muscle. So this is coming off of finishing skeletal muscle. There are going to be a couple of little similarities from skeletal muscle to smooth muscle, but there are some important differences that I will highlight and pick out. So when we talk about smooth muscle, the introduction to it here, looking at the location of smooth muscle, we're going to have the vasculature, the GI walls, the urinary system, the respiratory system, reproductive tract, especially like in the uterus, as well as in the eyes within the pupil. The iris, not the pupil necessarily, but the iris has two muscles which are smooth muscle and controlled involuntarily. You can see here in our vessels, especially in the arteries, we have this really thick tunica media where most of our muscle is going to be, and that's going to help regulate and control our blood pressure as well as blood flow to all the different areas of our body. Now, talking about what do most of these have in common, the big one, again, it depends on what your teacher wants to really highlight in there, but the big one is always going to be that these are all involuntary movements. These are all involuntary muscles, meaning that we do not have conscious control over their movement. We can't really control the dilation or the constriction of our eyes. We can't control our GI tract. We can't control our blood vessels like we can skeletal muscles. And also kind of breaking down smooth muscle, there are two sort of subcategories of smooth muscle. We have unitary, also known as single unit, and multi-unit smooth muscle. Unitary is also known as visceral, visceral meaning that with our hollow organs. So our single unit smooth muscles in the walls of the viscera, in the walls of those organs, has a stress relaxation response that permits the muscle to stretch, contract, and relax as the organ expands. Our multi-unit smooth muscle cells do not possess the gap junctions, and contraction does not spread from one cell to the next. That's the big thing that we want to see that's different, are those gap junctions. We saw the gap junctions in our cardiac muscle cells that allow the signal to transfer through. So we look at, for example, in the small intestine, or those hollow organs, we have these gap junctions which connect all of the different cells. And we'll talk about this a little bit more as well. We have these things called these varicosities from our autonomic nervous system. Rather than having a neuromuscular junction like skeletal muscle, we have these varicosities, which is how our signal gets transferred into the skeletal muscle. For our multi-unit smooth muscle cells, more like those are found in the eye, as well as some of the large arteries in the lungs. Mainly those three areas. These do not have those gap junctions, and they're separate. So this is a little bit more fine control, kind of more general control. So our single unit smooth muscles, this is general control, and our multi-unit is more fine control. Now, jumping into the anatomy, the smooth muscles are going to have, they're going to be single muscle cells, or they'll have a single centrally located nucleus. We look at skeletal muscle. Typically, we have they're all multinucleated. Cardiac muscle will have one or two nuclei within that. But our smooth muscle, we're always going to have one single centrally located nucleus. The shape of them are going to be spindle, also known as fusiform. The fusiform is kind of this almost football-y kind of shape. And they're connected to each other by these things called dense bodies. The dense bodies are the anchoring sites, kind of like how we looked at the sarcomeres are separated by the Z-disc, and the Z-discs are connected to each other in skeletal muscle. That's what these dense bodies, that's what their function is for. So the actin are attached to the dense bodies, like how in the skeletal muscle they're attached to the Z-disc. And we have intercellular bonds bind at the dense bodies in order to have a more forceful contraction. Also, it's important to note in the smooth muscle that our sarcoplasmic reticulum, whose main job is holding that calcium, is actually less developed in our smooth muscle. There aren't any T tubules. We have these things called caveolae. Okay, caveolae are these invaginations of the cell membrane, where the calcium enters the smooth muscle cell. And here's just a little bit of a definition down here of what those caveolae are and kind of their function. Continue with the anatomy. There is tropomyosin present, but there is no troponin. So this is a little bit different than the skeletal muscle. Skeletal muscle, we had the troponin, the three different parts of troponin bound to the tropomyosin, and then when the calcium would come in, it would all move and rearrange and shift to allow the actin and myosin to bind. But in smooth muscle, we have a little bit different process. So there is no troponin in our smooth muscle. As well, the actin and the myosin are not arranged into sarcomeres. Remember, the sarcomeres are what give skeletal muscle that striated appearance. So we don't have any sarcomeres, that same sarcomere structure in the smooth muscle. We do not have that sarcomere structure in the smooth muscle. The myosin filaments are all going to move in the same direction. And there's actually 5 to 10 times more actin in smooth muscle than there is in skeletal muscle. What this allows for is actually a much, I would call it a deeper contraction. So it allows us to contract a lot further. So whereas like skeletal muscle, we can only really contract about 30% of the skeletal muscle length. We can contract up to 80% of our smooth muscle length. So you get a much larger contraction with that smooth muscle. And we'll talk more about kind of how that contraction is also different. It's really cool how smooth muscle really works. So also comparing our smooth muscle to our skeletal muscle, we talk about cross-bridge cycling is slower in smooth muscle. So remember that cross-bridge cycling. This is kind of what we're talking about, from the high energy state, the power stroke that has it. This whole function, this is what's happening, how the myosin binds to the actin, you get the power stroke, and then it releases and resets. That's what we're talking about. So that whole cycle is occurring much, much slower in smooth muscles. And that's we're actually able to get a slower and a more sustained contraction and maintaining that tension over a long period of time. If we think about that, why would we want that to occur? We think about in the lungs, for example. Why do we want it to be either to contract or to move slower? I think when we breathe, we want to be able to take a really deep breath in and have the time all to let it all the way out. We don't need that really fast movement in the GI tract. We don't want to eat something and have it just shoot through our system. We need it to be able to move slowly and kind of get crushed and squished in order for our intestine to absorb the nutrients in it. So we need that slower, more sustained muscle contraction. Also the eyes. Like if we're consistently in a bright environment, we need our eyes to be able to be the pupil to constrict and to hold that constriction for a long period of time. And thanks to this slower cross-bridge cycling, some of those things we're able to do. So that cycle is actually 1/10 or up to down to 1/300th of the rate of skeletal muscle. As well, because that cross-bridge duration is prolonged, meaning that it's less often that we detach from the myosin and actin. First of all, that is due to there being less myosin ATPase activity. So remember, we need ATP to come in to bind in order to detach. So because there's less of the ATPase activity, that there's going to be less detaching of the myosin off of the actin. So that contraction and reaction occur at a slower rate. So just kind of the review. The functional characteristics of the smooth muscle, they differ in the excitation coupling, length, tension, relationship, energetics, and control of contraction. We'll talk a little bit more about energetics in a little bit below, but understand that if we are maintaining this tension by not attaching ATP, so ATP is not being used up, we're actually able to maintain this tension using less ATP. So our smooth muscle is actually going to use a lot less ATP than our skeletal muscle would. So it's much more efficient in that way. So now, getting into the actual contraction aspect of smooth muscle. We're going to see some similarities to the skeletal muscle, but I want to make sure we stop and point out kind of those main differences. So the way that it all sort of starts, let me get a good picture here, is it all actually sort of starts in this ECF. ECF stands for extracellular fluid. Out here in the ECF, we have a much larger concentration. It's more saturated with calcium. And that's going to actually cause calcium to come into the cell when our gates get opened, when the calcium gates open. When those open, calcium comes into the cell, and this calcium coming in also stimulates the sarcoplasmic reticulum to release calcium as well. This kind of combination, both of these releasing calcium, we'll call the calcium spark, is what it's called. I know Dr. Tate specifically doesn't talk, or he at least doesn't mention it in the PowerPoints themselves, but in case you ever get asked about that, that is what that is referring to, the calcium spark. Just the calcium, the initial wave of calcium coming in, and then activating the sarcoplasmic reticulum to also release calcium. So as the calcium comes from the ECF into the ICF, or the intracellular fluid, it activates a protein by the name of calmodulin. Okay, calmodulin is in charge of mediating the calcium regulation of a wide range of the physiological processes throughout eukaryotic organisms. Right now we're going to focus on that smooth muscle. So for the case of the smooth muscle, we get the calcium come in, more calcium is released from the sarcoplasmic reticulum, and the calcium binds to calmodulin and activates calmodulin. Now calmodulin is going to activate this other protein called MLCK, or myosin light chain kinase. If we think back to biochemistry, kinase enzymes are going to phosphorylate things. That's their main job. They do phosphorylation. So the MLCK, that myosin light chain kinase, is going to phosphorylate the light chains in the myosin head and increase the myosin ATPase activity. So if you remember, the myosin heads are going to release when ATP binds to them, and they're going to reset and cock and get ready and be full of energy in order to latch on and perform a power stroke. So that's MLCKs, the myosin light chain kinases. That's their job, is to prime all of our myosin. And then when we have our actin available, our myosin is then going to bind, and we're going to get a power stroke, get those cross bridges, creates that muscle tension, and so on. And that MLCK, as long as the MLCK is still active, it's going to continue to help in that process of phosphorylating the myosin, having them release and reset and continue to pull and cause muscle contraction. So there's a whole lot of words here. I'm just going to kind of start from the top here. We went through the whole process. I'm just going to make sure that I cover each of these each of these individual lines. So excitation, contraction, coupling, and contraction. So the excitation, contraction, coupling, where we have excitation, we're going to get the contraction, and those go together. We have excitation first and then contraction, and then we get the contraction after. So rise in that ICF calcium results in activation of calmodulin. The contraction occurs through interaction between the actin and the myosin, just like the skeletal muscle. The regulation of contraction does not depend on troponin. This is where it's different from skeletal muscle. But it does depend on the phosphorylation of the myosin ATPase. Let me talk about the control of the contractions are controlled through the ANS, or the autonomic nervous system. The autonomic nervous system is our involuntary nervous system, responsible for all the stuff that we do by ourselves. It's that fight or flight, sympathetic, parasympathetic nervous system. And the ANS has these little bulbs called varicosities. Varicosities are a series of neurotransmitter-filled bulges along an axon as it courses through the smooth muscle, loosely forming motor units, and a varicosity releases neurotransmitters into the synaptic cleft. I put a nice little picture in here to pull up. So we have these are what are varicosities. These are axon, and these varicosities, and those vesicles. We talked about vesicles before with neurotransmitters there. And these can actually be activated by a number of different ways. Either it can be a signal sent from the brain, or oftentimes it can also, and we'll get into more of the specifics of this, but it can be also other hormones. It can be due to actual movement. So like in the GI tract, stretch is a really big one. So when we eat a lot of food, it literally stretches the smooth muscle stretches our GI tract, which is then going to cause these vesicles to react and cause the muscle to move, to contract, to do what it needs to do. So there aren't any true neuromuscular junctions like there are in skeletal muscle, just these varicosities. So getting to that, the specific, we went through this already. We have that rise in the ICF calcium results in activation of calmodulin. Actually, I want to pause a second here and go into kind of how that occurs. So our smooth muscle also contains voltage-gated calcium channels as well as ryanodine receptors. Remember the ryanodine receptors that we talked about in skeletal muscles? Those are those mechanical receptors, and these are responsible for increases in the intracellular calcium concentration. The depolarization causes calcium channels to open, enabling the calcium to enter down its concentration gradient into the cell. And that's when we get the start. The calcium activates calmodulin. Calmodulin activates MLCK, myosin light chain kinase. MLCK phosphorylates the myosin chains. The capacity of the binding repeatedly, so it continues to bind over and over and over again. You get the sliding filament mechanism, and then it's stimulating the muscle contraction. The muscle contraction occurs. The relaxation, just like how in skeletal muscle where we pump the calcium out and everything kind of sets back into place. The calcium decreases, it gets pumped out of the cell and pumped back into the sarcoplasmic reticulum through our lovely calcium ATPase proteins. The amount of the activated calmodulin decreases. So because the calmodulin is no longer active, the MLCK is no longer going to be active. But this actually doesn't change the phosphorylation of the myosin light chains, of those myosin chains. So we need another enzyme is required for the phosphate removal of from the myosin. So remember, this is also one of the reasons why we can sort of maintain this contraction for a long period of time, because it's not really necessarily something specific, or I should say there's a secondary process required in order for the myosin to detach. So we have this thing called the myosin phosphatase. So the myosin phosphatase is going to increase. This is the other enzyme that is going to dephosphorylate the myosin chains. The cross-bridge cycling decreases, and this is also going to determine the rate of relaxation. So if we have more myosin phosphatase, we're going to have an increased rate of relaxation. And again, so this doesn't have to be released right away. We can have that held and sustained contraction for a very long time with the smooth muscle without using any additional ATP or additional energy. Getting into the membrane potential of our smooth muscle. So with contraction of the smooth muscle, there's the control of the contractions, like we talked about, is controlled through the ANS, through those varicosities without the true neuromuscular junctions. I kind of want to pause here and say that it's important to understand that there are different neurotransmitters, such as norepinephrine, acetylcholine, epinephrine, etc., like GABA, glycine, all of those in different areas. And they're all going to have they're going to cause a different response depending on the receptor that they bind to. The two big ones that I recall focusing on are the vasculature and the intestines. If there is another area that your professor wants you to focus on, make sure you learn those specific areas. But remember, the vasculature and the intestines were two really, really big ones. And also, you'll talk a lot about the vasculature and the intestines moving forward in future classes as well. So, talking about norepinephrine. Norepinephrine, and I'm actually going to even just draw a line straight through here because it depends on where it's binding. Norepinephrine, we have these different receptors in different parts of the body. We'll talk about like alpha and beta. In the future, you'll talk a lot more about these different types of receptors and kind of where they're located. But for right now, just the ones that you talk about here, those are the ones that you want to focus on. So for example, alpha-1 adrenergic receptor. So when we talk about an adrenergic receptor, it's a fancy way of saying epinephrine and norepinephrine. It's meaning that receptor, it gets epinephrine or norepinephrine binding to it. That's what adrenergic means, okay? So with norepinephrine, alpha-1 adrenergic receptors are most commonly found in the vascular smooth muscle, so the vasculature. And norepinephrine is actually going to stimulate contraction of that smooth muscle, activating it through the second messenger system, which results in an increase in the intracellular calcium, and then we get the whole process, the chemogulin, etc., etc., etc. With our beta-2 adrenergic receptors in the airway, actually stimulates relaxation. This is one of the ways to kind of remember this. We think about an EpiPen. An EpiPen is epinephrine. So our norepinephrine, actually, I believe an EpiPen is. Yeah, the EpiPen contains epinephrine. Epinephrine, norepinephrine are going to act fairly similar. When we inject epinephrine into somebody who's having an allergic reaction, If you think like a very severe allergic reaction, you typically see somebody, they're not able to breathe. And so actually injecting with epinephrine, activating these receptors, it relaxes the airways and also causes blood to flow quicker. It causes contraction. We'll talk a little bit more about that in the future, especially when you get into like pharmacology, toxicology, that course, as well as general pathology. We'll talk about histamine and kind of the inflammatory process and how that all works and how these different receptors respond to the different allergens and stuff like that in different parts of the inflammatory process. And now acetylcholine, as we had to talk about norepinephrine, now we have acetylcholine. So acetylcholine in the vessels, they tend to induce relaxation of the smooth muscle of the vessels. So relaxing the smooth muscled vessels. However, in the intestines, if you put acetylcholine or release acetylcholine into the intestines, that actually causes contractions. I'm not sure exactly how this professor taught this chunk here, but this is the simplest breakdown that I can give you when we talk about acetylcholine. This is the big one that I think is most important to remember, that the GI smooth muscle is contractile with acetylcholine. Because there are conditions where people can have not enough acetylcholine being released, and then they have a decreased movement of their intestines, and then they have decreased uptake of the nutrients, and they have other issues. Along the same lines of the control of contractions, so we talked about the norepinephrine, we talked about acetylcholine. There are other hormones, local factors, as well as stretch. Some of those hormones, norepinephrine and epinephrine, are cool because they can be both a hormone released in the bloodstream as well as a local neurotransmitter. So both of those they have that kind of added effect, which is cool. As well as estrogen and oxytocin. So hormones and local factors and the stretch, they can alter the affinity of the myosin light chain for the myosin light chain kinase. So different factors that stimulate the smooth muscle contraction. You'll learn a lot more about like the vasopressin and the oxytocin in Physiology 2, but just a little bit of information here. The vasopressin is aka ADH or antidiuretic hormone, and that works to help maintain our blood pressure, helps antidiuretic means that it's going to prevent you from your kidneys from filtering out a lot of fluids. It's going to hold on to the fluids. So its job is to kind of act on the kidneys to hold and to keep fluid in the body, to keep the blood pressure maintained. And there's something called pharmacomechanical coupling. This is going to occur when chemical signals change muscle tension through signal transduction pathways with little or no change in the membrane potential. So here are muscle tension. We can add and remove the different bits and pieces of this, so we can have hormones or local factors of stretch acting at different times where the membrane potential really isn't affected, but it does cause changes in our muscle tension. And that's what the pharmacomechanical coupling is referring to, where we have like the pharmacologic side of things, including the hormone local factor stretch, as well as even like medications can mess with this kind of stuff. Fake hormones, fake local factors, or lab processed. Can change the muscle tension. We think adenosine is one of those. So these are ones that do contraction. The vasopressin is contraction. Oxytocin, uterine contraction especially. That's what we'll call a feed-forward loop. We talked a little bit about oxytocin in Biochem 1 as well. And then the stretch reflex, where we have mechanically gated calcium channels, which will open just like in the intestine, is a good example of that. Some of the factors that stimulate the smooth muscle relaxation, and we talked about how using an EpiPen for allergic reaction, but also adenosine. So think back to biochem is when we're more active. We get more adenosine being released. and that can cause, that's why often, like, when you go running and you start moving, there's a couple of reasons, a couple of reflexes and stuff involved in this process, but when you really start getting active and moving, a lot of times suddenly you have to go, the urge to go to the bathroom, when you start running, and part of that is due to this smooth muscle relaxation, caused by the different factors like adenosine that occur in the bloodstream when you start to exercise, as well as nitric oxide. So nitric oxide, this has a lot to do with our diet. So we have a diet higher in nitrates and nitrites, like from the dark green leafy veggies. Nitric oxide can also be given artificially through either pills or through a patch. And it's a treatment for high blood pressure, because the nitric oxide causes the smooth muscle in the arteries to relax, which will lower our blood pressure. The length-tension relationship in smooth muscle. So tension development and resting length are not directly related. There are functions over a large range of lengths, and a stretched muscle will adapt to the new length. This kind of goes back again to what we say, how they can hold tension for long periods of time, and that doesn't bother. They don't fatigue in the same way that skeletal muscle does. See, where our total force versus passive, where here our total force, passive and active, our length by force. Our total force can keep going up and keep going up, unlike our total force from our skeletal muscle. This just gives it the ability, first of all, it allows it to retain the capacity to contract, as well as still function over a wide range of lengths. Even think about the uterus, think about the stomach, think about the bladder. These things, which are designed and built to truly expand, like the uterus expands immensely, but it's still able to function and produce the total force that it needs to produce. We've touched on this. This is just defining it, the name, where that prolonged contraction with MLCK and the myosin phosphatase decrease in activity. This is where the tension is maintained. We just call it the latch state, when the smooth muscle is maintaining the tension and holding, and there's just the myosin heads just aren't being caused to release from that, and that can be held for days or up to weeks, depending on what's going on. Here's a little bit more in-depth, kind of step-by-step, kind of how that happens, if you want to pause and go over that as well. Membrane potential. So the resting membrane potential for smooth muscles between -50 and -60 millivolts. If you think the neurons are closer to -70, skeletal muscle more like -80, cardiac as well, cardiac and skeletal both more closer to like -90, in that range. So action potentials are rare in multi-unit smooth muscle, and the action potentials in the unitary smooth muscle occur as normal in skeletal, like they do in skeletal muscle. So we have these two things are called spike potentials, as well as an action potential with plateau. So these two different types of action potentials. The spike potential is the one that's similar to skeletal muscle, and that's seen in the unitary smooth muscles, where you get the signal, it spikes up and comes back down. With the plateaus, this starts like the spike, but repolarization gets delayed. This is helping with the prolonged contraction that occurs in the different organs that we want to have occur. There's lots of cool stuff here. This is my other physiology information and stuff that I have. I feel like this does go into a lot more detail of everything, but again, it's previous professor did lecture a lot more and had a lot more material than the current professor does. So I don't want to give you too much in here that might confuse you. This is kind of repeating a little bit where the calcium is important to generate action potential. We know why, going from the ECF to the ICF, activating the calmodulin, all that sort of good stuff. The smooth muscle contains more voltage-gated calcium channels because the calcium is required to start this whole process, as well as part of that maintaining of the plateau. There are few voltage-related sodium channels, which is responsible for prolonged plateau observed in smooth muscle action potentials. The calcium voltage-gated channels open slower than the sodium channels, which contributes to the membrane potential and the process of the muscle contraction, and the longer action potential duration leads to, not sure what that is supposed to be saying. The longer action potential duration leads to, my guess is supposed to be talking about the longer contraction time. We'll talk about this in one more sec. Pacemaker potential. Actually, we'll talk about it now. So we talk about pacemaker. This is what we're talking about. So cardiac muscle and some smooth muscle show pacemaker activity. So pacemaker activity is where a few specialized cells, they act as their own, they bring themselves to threshold without receiving a stimulation. So there isn't really a stable resting membrane potential. So as soon as it reaches its resting membrane potential, you can have leaky channels or different things in these pacemaker cells that slowly begin bringing it back to threshold to cause a rhythmic depolarization. So it's a rhythmic action potential leading to a more rhythmic muscle contraction. The cell gradually depolarizes, reaches threshold, and fires an action potential with no external stimulus needed. So this is how, like, in the stomach, in the intestines, rather than the brain constantly sending signals, it's able to sort of do this on its own and have a nice steady pace of peristalsis. It's called peristalsis, is the movement through these hollow organs. So, like, kind of, I think he has a picture over here. We'll get to this in a sec. The slow wave potentials in the digestive tract. Due to these pacemaker potentials, we get these slow wave potentials. The oscillating membrane potential without external stimulus. The intrinsic property of smooth muscle, it's not considered an action potential. This does not stimulate presence of spike potentials. So the cyclic opening of voltage-gated calcium channels leads to depolarization, followed by the potassium channels opening, you get repolarization. And depolarization brings membrane potential closer to the threshold, increasing the probability of generating an action potential. Apologize, two different things. Pacemaker potentials are one thing. These slow wave potentials are something else entirely. Let me find the... They're both in the same boat. They kind of happen by themselves. The slow waves and the pacemaker potentials both are occurring without external stimulus. But again, with these slow waves, they're going to kind of, you get these amplitudes and these amplitudes and these amplitudes. And when it reaches threshold, we're going to get openings of the calcium channels. Those calcium channels are activated, resulting in calcium influx and an initiation of the motility, especially in the GI tract. And from the muscle stretch, we touched on a little bit. So when the unitary smooth muscle is stretched, spontaneous action potentials are generated. You get normal slow wave potential, opening of the stretch channel, calcium coming in, all this stuff. And the automatic waves are generated. You get peristaltic waves. Peristalsis is the movement of stuff through the muscles. So you get the posterior portion contracts, and it kind of like squeezes it along. You get relaxation at the site of the peristalsis and forward. And so it kind of contracts here, and it's going to slowly contract, and push what's called a food bolus forward. So that is all the information about smooth muscle. A little bit longer than I would have liked, but lots of like kind of little details in here. Most professors in this most of the time, they don't hit smooth muscle super hard as far as examinations go, but it is important to really to understand this, and it's kind of an introductory topic here, especially for future tries. You will see a lot of stuff in your pathology courses and stuff about smooth muscle and its involvement and kind of how it does its thing. So very important to know and to understand now to set you up for the future. Thank you for watching, and I'll see you in the next one.